Okay, welcome everybody to the second seminar, ACOM seminar of this week. Today we are hosting Jose Fuentes, who is a professor in the Department of Meteorology and Atmospheric Sciences at the Pennsylvania State University. Jose has been at Penn State since 2009. Before that, from 1995 until 2009, he was a professor at the University of Virginia, including holding the position of Cavalier's Distinguished Teaching Professor. From 2005 until 2007. Jose received his master's and PhD from the University of Guelph in Ontario. I had to learn how to pronounce that today. Jose has received numerous awards, including the Charles E. Anderson Award from the American Meteorological Society, in which he is a fellow, the State of Virginia Outstanding Faculty Award, and the George H. Delkey Jr. Research Award at Penn State. He is a current editor of the AGU publication EOS and Agriculture and Forest Meteorology. He has also served as an editor for JGR and ACP. Jose's research interests include surface-atmosphere interactions, including trace gas fluxes and chemical reactions in the boundary layer. As an educator, he also involves many students with field research and disseminates knowledge to students and the general public, including underrepresented groups. Today he will talk about floral scents emitted by vegetation, pollinators, and atmospheric chemistry. So please welcome Jose.
Thank you very much, John, for that introduction. And also, thank you very much for coming to today's seminar. And I really welcome the opportunity to share with you some results that I have derived over the last few months. I have been spending part of my sabbatical here at the National Center for Atmospheric Research with colleagues John Orlando and Geoff Tyndall. And today I'm going to kind of summarize some of the ideas that I have learned in the last few months. I'm going to speak about a topic that I believe most of us in this room care deeply about, and this is the issue that pertains to the emissions of these floral volatiles that plants and flowers produce to attract the pollinators, so that these pollinators can in turn give us the beautiful apples, oranges, strawberries that we all love to eat, and also the tomatoes that we like to have, and the cucumbers and the squash. And this process of insects being attracted by the plants, particularly the flowers, is so important. And if you basically tally up all the services that these bees produce worldwide, they give us about $225 billion US per year. When I mentioned this figure to Paul Shepson, he said to me, "Oh, we can fund NSF for 25 years, he said to me, which is a staggering figure. But one of the ideas that I want to tell you here today is that as soon as these gases are leaving the flowers, they begin to be attacked by oxidants in the atmosphere. In other words, this research here requires the knowledge of emissions, turbulent transport, and more importantly, the reactions of these gases. As you can imagine, this is a very interdisciplinary type research, and it involves the participation of colleagues from various fields. And I have been very fortunate to interact with chemists such as John Orlando, Geoff Tyndall, Ty Rolston from the University of Virginia, who is an insect ecology, Marcelo Shamecki, who is an expert in large eddy simulations. And so for today's talk, I am going to kind of share some results with you about the summary that I have been able to produce this fall as part of the plant-insect interactions and the associated chemical cues that insects actually need to locate resources. Also, I will apply some numerical models to determine the chemical destruction of these molecules as they travel from flowers to downwind places. And I will also use that information from these models to determine how much the modified scentscape actually can produce some changes in the foraging strategies that these insects actually have to pursue in polluted air masses. And then finally, I would share with you some ideas that I have been doing in the laboratory lately, in which I interrogate the insects. I ask the insects virtually whether they like polluted air or not. So this is something that I hope you will be able to learn for the first time. So just to start, when we humans want to get something or want to get to somewhere, we always are using visual cues. So for instance, in my case, if I want to locate the main building of NCAR, I have to look for the features of that landscape. Or if I want to find the physical address of a place, I have to use my visual cues to locate that. Or if I want to locate coffee somewhere in San Francisco for the Bumblebee coffee, or in Penn State, the Starbucks we have there, we are always using visual cues. In the case of the insects, most of them cannot see from afar. In fact, most insects can only see within a distance of 10 centimeters or so that they can discriminate the most beautiful flowers that they can visit. Instead, when these insects are actually foraging across the landscape, as they navigate through the landscape, they are actually the best chemists we have in the planet. So what they see here is, I am plotting this chromatogram in which each little peak that you see here represents a different molecule. So we call that the retention time. This is actually what we produce from the gas chromatograph or the mass selected detectors. And then these insects can detect not only the identity of those molecules, but more importantly, the amounts or the number of molecules that are impinging on the antenna that they carry. And then they can fly against the gradient, and that's how they locate the flowers. So in our case, what we do, well, the chemist will tell you, well, these blue peaks that you see here belong to the monoterpenes that a lot of chemists have studied in many years. And here we have the aliphatic acids, we have the terpenoids with the alcohols, and then we have many aromatic compounds. So these plants, these flowers, and the insects basically communicate that way. And so in my own way of defining what I mean by plant-insect interactions, what I mean is that in the case of the flowers, this is the most common feature that we all relate to when it comes to plant-insect interactions. Those flowers release these molecules. And please note that these are really big molecules. And then the bees detect the scents, and that's how they can really locate the flowers. But the other component of this issue of plant interactions is that when we have insects such as the herbivores that are actually munching on the foliage, the plant will immediately release a burst of these molecules to advertise that it's being attacked. And then the enemies of the herbivores, that's actually how they can locate their food. And then they can come and actually they have lunch with the herbivores that are actually munching on the foliage. The same thing when you have the aphids that are basically sucking on the foliage, and then you have the beetles that come that way, and the beetles actually do this way. So the story I want to kind of convey today is that as these molecules are being emitted from the plants, and if they are released in environments where we have plenty of oxidants such as ozone, hydroxyl radical, and nitrate radical, then these molecules are going to be degraded. And there are two things that can happen. One is the actual amount of these molecules will be decreased, and the other one is that the chemical composition, the blend that I showed you earlier, is going to be changed. And so this is really crucial then for these insects to locate the flowers, because these flowers produce the scents for the insects to detect or to be attracted to the plants. And these scents, unlike us, you know, our visual cues are obstructed sometimes. In the case of the odors, they can travel anywhere in the environment, and all these molecules that are produced by the flowers are actually conveying some information to these insects. These molecules tend to travel anywhere between 100 to 1,200 meters, depending on the reactivity of these molecules. So in the insect world, you can classify insects as being a specialist, meaning that they are associated with an individual plant that releases a given type of molecule, or you have insects such as the butterflies that tend to be generalists. They will go for a broad range of volatile compounds. And so ordinarily, the way we do these studies is that first, if we want to identify the chemical species produced and emitted by these flowers, we have to build these environmentally controlled chambers. And then we put the flowers in there, and then we know what we put into the chamber, and we know what we get out of the chamber, and then that's how we can calculate the flux of the gases produced by the flowers. We use the mass balance approach to calculate the flux of the flowers. So we need to know the flow rates, we need to do the chemical species going in and out of the chamber, and we also need to know the active biomass in the chamber. And that's how we can calculate the actual flux of these gases. I probably failed to say a moment ago that if we take an inventory of all the molecules produced by the flowers, flowers produce approximately 2,000 molecules. And when I began this research, I was astonished that less than 10% of those molecules have been studied for their kinetics. When I told this story to John Orlando, he looked at me very suspiciously. He said, the chemists know everything. How come we don't know these molecules? And then I began to dig deeper, and then, believe it or not, the people who really study these molecules they only know what molecules plants produce. And it is mostly the chemists who really want to put some order into this research and produce, for example, families of these compounds. So John Orlando and Gio Taglio and I have been working on this idea in which we are trying to come up with a family of these compounds. And most of you in the audience, I think, will probably recognize this family, the monoterpenes, that have molecules that have 10 atoms of carbon, 16 atoms of hydrogen, and you have these two molecules here. That's an example, the ozamine and the terpenoline right there. And one feature that you really need to recognize in this molecule is the ubiquitous presence of these double bonds, which means that these molecules are very reactive. That's the key attribute. Then when we move on to the sesquiterpenes, we have bigger molecules and therefore more likelihood that these molecules will react with oxidants in the environment. And then you have the humulene. You see how complex those molecules are. These are really very expensive molecules for flowers and plants to produce. And then we have what we call the fatty acid derivatives, which are hydrocarbons that have oxygen in them. Two examples include the 2-methylbutanol, you can see the picture here, the 2-hepanone molecule you see there. These molecules actually are produced when the plants are stressed, and this is a signal that they send out. And then we have a very broad range of molecules in the benzenoid family here, and an example of that is basically this formaldehyde that you see here, the benzene ring. A few years ago, John Orlando and I and Geoff Tyner were in the laboratory, and this molecule here, it smells like cinnamon buns. That's the sense that you get from this one here. And then we have, this is eugenol. This is a molecule that is abundantly produced by all the petunia flowers that we actually grow. And then finally, I have these two new families of molecules here, not new, but two other families, nitrogen-containing compounds, such as the indole that you see here. You can see the nitrogen in this molecule, and this molecule is abundantly produced by the squash flowers. And then you have these two, this other molecule, imidazole, this one here, I think I'm mispronouncing this molecule, which basically has 2 nitrogen and then it has 4 hydrogen. And this molecule is actually produced by the cobra lily. And then we have this very peculiar compound, the sulfur-containing compounds that are produced by the dead horse lily. These two flowers that you see here, we call them deceitful flowers because they release compounds such as dimethyl sulfide or dimethyl trisulfide. These are compounds that basically give you the sense that the meat is decaying. When you smell these flowers, you really want to get away from those flowers. And what they do is that they attract the flies. The flies get into the flowers and pollinate the flowers without them, the flowers, providing anything to the insects. That's why we call them deceitful, because they don't provide any rewards. Whereas these are the flowers here, they provide rewards to the insects. The more nectar, the more pollen that they can provide to the insects, the more insects that visit. No wonder why only flies can visit these flowers. Okay, so I need to take a little break here and tell you a little anecdote. Teaching and research is perhaps the best job there is. When I was teaching, I teach a course in air chemistry. An insect student came to see me, and he said to me, I heard that you are a very crazy teacher, and I want to take a course with you, but I have no idea about turbulence or equations, none of that. I only know how to say hello to the bumblebees. Can I still take the course? And I said, sure, take it. And then he taught me a little bit about the entomology. And at that time, when he was taking that course, there was a report by the National Research Council of the United States in which they included a figure something like this. And in this figure, you have on the y-axis the number of big colonies, and on the x-axis, you have the number of the colonies with time. And then even there was an article in Nature, and at that time, for a long time, entomologists have been saying that we have the colony collapse disorder. The colony collapse disorder is something akin to basically saying we don't know what's happening. Basically, that's what it is. And so I became interested because I knew a little bit about the chemistry. And then the more I became interested in this topic, then I learned that the honeybees, this is the honeybees, are not the only insects that are in trouble in the world. So we have the honeybees are in trouble worldwide. The bumblebees, which are cute because they wear the jersey of the soccer team from Brazil, they are in trouble. If you go to the San Francisco area, they are almost ready to go away. And this is really sad. And then we have the solitary bees that are in trouble. And then you probably have heard about these beautiful butterflies that are also in trouble, mostly because their habitat in Mexico was being destroyed, but the president of Mexico last year restored that habitat. And so we have basically learned over the years that there are many reasons why our insects, our pollinators are in trouble. One reason is that we are eliminating or reducing their native habitats. We are just eliminating. If you go to where I live in the eastern part of the United States, we are like there's no end to tomorrow. We are increasing the fragmentation of the habitat where these beautiful things should survive. And then you probably have heard about this. There was a science article last December that said that the most important aspect that is killing our bees is these compounds here, the neonicotinoids, that are basically decimating our bee population. And of course, we have some diseases. Any biological organisms will experience that. And so when I began my research, I said, wait a minute. If these molecules are so reactive, there has to be a component related to the chemistry of these molecules in the air. And then I posed this hypothesis in which, I said, wait, the modification of the bouquet of flowers that these oxygens produce must be producing some stress on the insects. And I will provide you some evidence towards the end of this presentation that that is indeed the case, that is happening in some jurisdiction. But very briefly, though, these floral scents are oxidized by ozone, hydroxyl radical, nitrate radical. These reactions produce these compounds, and John Orlando and Geoff Tyndall have been kind of postulating that perhaps, some of these new molecules can also serve as the cues that these insects need. And we're doing laboratory studies right now to learn that. But the most important thing that we can do now is to investigate how this chemistry impacts the integrity of that bouquet of scents that the flowers produce, plus the strength. The strength to me is like saying if the flower produces 100 molecules and only 5 reach to where the insects are, then we have changed the strength of that source of compounds. And I underline an ozone here because most of us in this audience have probably invested a substantial amount of time to study the chemistry of ozone. And all of us, I think, should be proud of the fact that in most places in the United States, the ozone has really dramatically changed over the last 25 years in the United States. And what I have done here, this is a very simple plot that I created for the ozone levels in all the stations in the mid-Atlantic region of the United States, including Virginia, Maryland, New Jersey, Delaware, and those places. And what I want to show you is that when you go way back in time, I only chose 1998, what you can see is that in the early part of the record, we used to have very high ozone levels. And also, the ozone levels were almost close to zero. These are the minimum ozone levels. But if you look closely towards the end of the record, which ends in 2012, then the minimum ozone has increased. And we know the reasons. All of you here probably know the reasons why. And so what that means then is that the distribution of ozone over the last few years, for at least the last decade, has changed from this distribution that you see here, where we basically had some very high levels here in the high range of ozone. But when you look at the most recent one, you can see that the ozone levels in the 30, 40 parts per billion are more frequent. And so when you would integrate that influence over time, of course this is going to create some environmental problems for our pollinators, and I will show you in a moment how. And then Owen Cooper, who is with NOAA here, published this very nice article in Nature about 6 years ago, in which he analyzed the data for the western part of the United States, for the layer from 3 to 8 kilometers. And you can clearly see a very nice trend in the ozone above the atmospheric boundary layer. And Owen and his colleagues really told a very beautiful story, because they were relating these ozone trends to the long-range transport of ozone, and they ascribed these ozone levels mostly to the long-range transport coming from Asia. So with this background, what I want to tell you then is to go back to the idea that we need a lot of new information, but principally about the kinetics of these molecules, so that we can understand the extent of the influences that oxidants have on our pollinators. And that chemistry is needed because we need to understand the degradation of all volatiles, not just the 10% molecules I show you. We need to know the new chemical species that are produced, and we need to know whether they serve as cues to the insects. And then we, more importantly, need to investigate whether this modified senscape is indeed modifying the foraging patterns that the pollinators have. And here, this is the picture of John and Geoff Tyndall's laboratory, where they have been helping with some kinetic studies. I think we studied one molecule already, alloocimine. And what I want to tell you is that, we need to know this chemistry of gases such as beta-ocimine, beta-marcin, linalool reacting with ozone. I left out the other reactions. And what I wish to illustrate with this slide is that these reactions basically produce common products that you see here. And right now, we are trying to study whether these other common molecules, such as the 4-methyl-3,5-hexadenal, are in fact being used by the insects to locate at least the plume of this scent. So that's really important. And so while I've been here at NCAR, I've been trying to understand, based on the availability of the kinetics data we have, and also using some theoretical models to estimate the kinetics of these molecules so that we can understand what their lifetimes are in the environment. And so in this slide, what I'm doing is plotting on the y-axis the lifetime in hours, and on the x-axis I have the ozone reactivity expressed in volume per unit molecule per unit time. And then I color-coded these molecules here. The blue are the monoterpenes, the magenta here, or this is actually purple, is the sesquiterpenes and the fatty acids and the benzenoids. I owe you the sesquiterpenes because they are way out there here. And I owe you the benzenoids because they are part of here, but I will highlight them to you in a moment. And I basically calculated this lifetime for 40 parts per billion of ozone and the mixing ratio of 120 to illustrate, that these are the levels that we still see almost everywhere during the summer. And you can see that these molecules that are in blue, they are all here. They only reside in the environment for just a fraction of an hour. So if you have a very gentle breeze passing by the flowers where they are produced, these molecules only travel a short distance, which is the distance I showed you earlier. And so we are using this data to understand how the bouquet of floral volatiles that are produced by flowers such as the petunia here. I have the petunia. This is the emission rates of these compounds that you see here on the x-axis. and the emission rate is microgram of that compound per dry of the floral weight and per day. And I basically organize this chemical species in alphabetical order, if you like. But what I want to show you is that when these flowers produce these compounds, they are producing a bouquet, a blend of these compounds, and that's what they have. And so you can see that in the case of the petunia, they mostly release benzenoids, whereas in the sunflowers, there is a mixture of monoterpenes, benzenoids, and even some fatty acid compounds. But in the case of the canola, which I wish to note that the low range here for the emissions is actually much higher than the other ones, then you can see that most of the compounds are actually in the monoterpene family. So if I were to behave like a chemist, I would say, oh, I can sense some degree of reactivity for these three bouquets that you see here. In fact, that's what we have done. We calculated the hydroxyl radical reactivity for the bouquet. And what I want to tell you is that you can see that the reactivity is very dependent on the type of compounds that these flowers produce. So in the case of the petunias, they go to 10 to the minus 4, 1/2 reactivity to 1, and then whereas in the case of the canola, basically is 10 to the minus 3. So the canola then is the reactive bouquet, the sunflower is the mid reactivity, and this is the low reactivity scale. And that all depends on the type of molecules that are produced by the flowers. So what I have done, and what I'm going to show you, is to basically integrate all this information about emissions, about chemistry, and the turbulent transport in some numerical models to learn basically the extent of the degradation of these molecules. And so this is basically the framework of the research, that I'm assuming that the wind is blowing away from the upwind region to the downwind, and therefore that those air parcels or air masses are being or have oxidants such as ozone, and those oxidants then will change the amounts and the composition of those floral scents. And then the insects that are downwind, they have to do the foraging to locate the flowers. And then we need to calculate what that foraging time is as a function of distance from the source and also as a function of the oxidant levels in the air. And also we are estimating the visitation rate. These are theoretical calculations. And so I'm going to share with you the results of two types of models that I have applied. One is a Lagrangian type model, very simple one, that the ecology colleagues actually like to apply. And then I'm going to also share with you the results from a large eddy simulation. So in the case of the Lagrangian model, we simply calculate the emissions of these compounds as some basal emission rate, and then we estimate the influence of the temperature on the emissions. It's very simple. I think for those of you who do the isoprene or monoterpene calculation, it's very similar. And I extracted this from the literature. And then in terms of the turbulent transport, what I do is that I apply a 3 Lagrangian dispersion model, and here is the source where I got the model. Basically, in that model, I can calculate the change in the movement of the air parcel. I release simultaneous parcels, more than 1 million at a time, and then I basically calculate the position of those parcels at any given time. So I will explain in a moment what this coefficient is, these two coefficients. And so I can calculate the displacement of the parcel by knowing basically the wind speed, the mean wind speed, and the fluctuation of the wind speed, and then that's how actually allowed these air parcels to go from the sources to downwind regions. And then these coefficients you see here, this coefficient is the memory term that tells me something related to the turbulence, and then this term is expressed in terms of the Reynolds stresses and also the rate of turbulent kinetic energy dissipation, as well as the mean shear that we have in the flow. In this case, I only apply this to about 10 meters from the surface. And then the B coefficient here that's the diffusion term that allows me to determine how rapidly these air parcels are diffusing throughout the landscape, and I use the concept of the turbulent kinetic energy dissipation rate. And then this W term that you see here, that's the one that tells me how about the stochasticity of the turbulence in the environment. So I'm assuming a variance of the time that I use, and then I assume that the turbulence for these calculations are Gaussian. In terms of the chemistry, well, I apply a very simple model here for the chemistry. For example, when I have linalool, I allow linalool to react with ozone, nitrate radical, the hydroxyl radical, and then I get the products. I don't really calculate anything about the products. I just want to know the rate of change of destruction of that compound with time, and that I solve this differential equation in my model. For the Lagrangian model, which my colleague Marcelo Chamecki is doing with me, so we basically have here the simple equations. This is basically to tell how fast the areas are moving. And so here you have the advection term, here the pressure gradient, and here you have the turbulent transport, and this is the subscale grid of the turbulence that we have here. For me, though, this is the most important equation right here, because this second equation is the one that allows me to calculate the variability of these gases in terms of the advection term, in terms of the movement of these molecules in the subgrid scale that you see here below the grid of the turbulence, and then I have the emissions, and then this solves the chemistry. And then we use the continuity equation. This model has a domain of 1,000 meters. I occupy the first 200 meters with the flowers, and we go from the surface to about 50 meters. So the boundary condition is like that. Then we use cyclic or periodic boundary conditions. And then finally, just to end this part of the atmospheric modeling, we have considered various scenarios. Scenario 0 means no oxidants in the atmosphere. Scenario 5 means 120 parts per billion of ozone, 0.8 parts per trillion of hydroxyl radical, and 5 parts per trillion of hydroxyl radical. There is one critical element in all of this work. And this critical element relates to how quickly these insects move in the environment. That's a very challenging thing to do. In the last year or so, I've been working with my fellow ecologists. And what we do is that we put instruments on the bees. This is true. We put instruments on the bees. And then we ask the bees to locate the flowers under different variations of oxygen levels in the landscape. So just to make this very brief, this is called a transponder, and we use harmonic radars to find the location of the instrument at the frequency of 1 hertz. And then what we have done, and also others have done that, is that we have constructed these probability distributions that tell, the length of the flights and the minimum step that they take to move from one place. So, and then we have this exponent here, and then that's how we model the displacements of these insects in the environment. And the other element is that we need to know what the detection threshold is for every molecule type. According to some research, those bees require as few as 6 molecules impinging on their antenna to detect or elicit a response from these volatiles. Okay, so the results. This is when I ran my Lagrangian model with no chemistry for the compound beta-ocimene. What you're seeing here is the discrete location of the sources of the beta-ocimene, and then here's a location of a hive of insects, and then we allow turbulence to disperse these compounds across the landscape. So here you have the vertical dimension there. And so this is the work of turbulence. But if I now add ozone, or use 120 parts per billion of ozone, then you can see that hardly any of those molecules reach the hives of the insects. So under these conditions, these molecules travel no more than 300 meters away from the sources. And this is one of the key questions that we want to answer, as to how much these oxidants are, in a way, degrading the strength of the source. And when we use the outputs from the large eddy simulations, so here we have the molecule beta-marcin, and then you have, in this domain here, you have the crosswind, downwind, and here's the vertical going from 0 to 50. So these plumes that you see here were created for the conditions when we have no pollution, no oxidants in the environment. And when you see down here, this is when we had basically the 120 parts per billion of ozone. And you can clearly see how much change there is in the distance that these molecules travel simply because we have the presence of oxidants. So that's in the case of the marcin. I have another result here that is for one of the reactive molecules, the beta-caryophyllene. And when you have beta-caryophyllene in the environment, you can see that it only travels just tens of meters away from the source. And part of the reason is that the molecule beta-caryophyllene is very reactive indeed. And so it's modified. When I show the results in a different orientation to look in the vertical, this is what you get for beta-caryophyllene. You can see the plumes, which is very characteristics of the doing of atmospheric turbulence. And you can see here too that these molecules they don't travel more than 10 meters away from the surface. So they are being oxidized by the presence of these compounds. And in the case of the beta-caryophyllene, the results are even more conspicuous because these compounds basically don't leave or don't travel too far away from the source. And then the last one set of results I want to show you is that when I examine these results from calculating the fraction of the compounds that are traveling as a function of distance, and then I have the various scenarios, and then I also include the effect of turbulence, friction velocity of 0.1 and 0.2, you can see that when we have turbulent conditions, then the molecules travel faster. And when we have light conditions or very reduced turbulence, then these molecules, by and large, remain very close to the source. And then in the case of the beta-caryophyllene, as I mentioned earlier, these molecules basically don't leave the flower beds because they are reacted, basically. They don't travel far away. Earlier, I think I mentioned to you that I wanted to illustrate the effects of both the changes in the strength of the source, which is this one, and now I want to show you the results in the changes of the composition of these molecules. So first, I want to illustrate that on the top panel here, I am producing the results for these molecules, linalool, beta-ocimene, marcin, alpha-pinene, beta-caryophyllene. So we have no effect of pollution here. And then on the x-axis, you see the distance traveled by the molecules. And this is the scenario with 60 ppb. So there aren't many changes in here. The only change is that this molecule, beta-caryophyllene, as I mentioned a moment ago, it only travels a short distance, even for 60 parts per billion. When I go to the next level, when I now plot the level of 120, that's when the whole composition of the bouquet has changed. Instead of having, for example, the ocimene being the dominant scent in the plume, all of a sudden, beta-pinene is the dominant species. Instead of having beta-caryophyllene in the initial bouquet of flowers, the caryophyllene is gone after it travels 100 meters from the surface. And these are the key results that actually can confuse the insects. And so we have used the insect movement data to calculate the foraging times that these insects will experience in an environment that, where their scent plume has been modified. So on the top axis, that's when we run the model with no oxidants. And on the x-axis, what you see here is the scent detection threshold, because not all the insects detect the same number of molecules with their antenna. So if I can randomly pick a number here around 100, right? So the insects in unpolluted environment will take probably 40 minutes to locate the flowers, but when you go into this one, it's 150 minutes. So that has changed very dramatically. And we have done the same calculations doing the LES, but I will skip this because I really want to highlight these experiments that we've been doing in the laboratory, where we have been asking the insects, such as the strip cucumber beetle, that basically goes after the indole in the squash flower that I told you earlier. I asked this beetle, do you like polluted air? Yes or no? And then I use these tubes, I put the flowers in here, and then I put one stream of that smelly. This molecule here smells like human manure. So I don't like to work with that molecule. So it's not like cinnamaldehyde. John and I love that molecule. And so we passed this through the reaction here. We reacted, and then we have all the equipment, the ozone generator, the ozone monitor, and all the gadgets that you need in the laboratory. And here's a picture of the gadgets. I put the insect here, and then the insect had to crawl in there, and then make a choice to go for the polluted and the unpolluted air. You can see the flower here in the environmentally controlled chambers, and here are the results. On the x-axis, you see the ozone mixing ratio in parts per billion. On the y-axis is the percentage of the choices. We asked 100 beetles whether they like polluted air. And when they reach this magic number, which some of you know, 80 parts per billion, they cannot distinguish the scent anymore. Beyond that point, they just couldn't. They always went, for the unpolluted air mass. And to me, that's a really, really important result. And we are doing similar work in the laboratory, which we haven't analyzed the data yet, to demonstrate that other insects are more sensitive than the beetles in order to actually detect these modified scents. So I want to kind of then leave you with these concluding remarks, and I hope that the results I show you give you the evidence that when we have elevated levels of oxidants such as ozone, hydroxyl radical, nitrate radical, they do those two things: reduce the strength of the source and modify the composition of the bouquet of scents that flowers produce. And also, these scents the reason is that they confuse the insects is that they don't travel that far. So as a consequence, it's like, you and I looking for a place to find, right? But if we have no cues, if our glasses are all destroyed, or say that John Orlando asked me, go and buy me a cup of coffee at the local Starbucks, but I'm going to blindfold you first, and then I will go and find the cup of coffee. I will have a hard time actually finding that Starbucks like he wants. That's exactly what is happening here with the insects. We are confusing them because of that. And so this is really, really important. And I think that these pollutants have the potential then to perniciously alter not only the pollinators, but all the insects that require these scents to actually find food. And I want to kind of also tell you the broader impacts of this research. It's really important to understand these processes because 70% of the crops that humans cultivate need pollinators in order for them to produce the food that we all need. I already quote you the figure, that came out. That's the figure of $225 billion US. That's a figure for 2009. It's probably more than now. And probably some of you may think that this only happens in the polluted places such as Los Angeles, Washington, D.C. I am here to tell you that there are places now in the world where people have to hand pollinate the apple orchards. Imagine you and I pollinating our tomatoes, our apples. That's really something. And you may remember this little story here that in 2008, the United States Department of Agriculture, for the first time ever, allowed the importation of honeybees to come into the United States from Australia to pollinate the almonds in California because they did not have the bees to pollinate the almonds. So this is a really serious issue, and I think that if we in the atmospheric science community have a unique opportunity to make a contribution with our fellow ecologists, because we know our chemistry, we know our turbulence, we know our numerical methods to kind of help understand these processes that have very broad ramifications. And I want to thank you for your attention. I should probably say that my research is funded by the National Science Foundation. Also, Penn State has given me some money to do this kind of work. And I thank you so much for your attention.
So thank you for that interesting talk, Jose. So we're going to open this up to questions. And if nobody wants to start first, I'll start first.
Go ahead.
And so one of my questions is, if you go to an area that is in an urban area, since the pollinators are attracted to not only one scent, but a bouquet of scents, have you considered, or has anybody considered, the impact of mixing in urban influences, let's say benzene, to the floral scents or toluene or things that you would see in a more urban environment in the absence of ozone, and the same concentration of what they're looking for is the same, but because the presence of this other solvent is around, it confuses them. Has that been considered?
No, I don't think so. That hasn't been considered. This past July, I published an article which received a lot of media attention. And I was telling John Orlando this morning that I received a phone call from a team of high school students in New Jersey. I love interacting with young people because they are the future. And they told me, we read your article, but we couldn't really understand what you were saying because your article was full of equations, a lot of chemical reactions. We don't want to know about this. Tell us what we can do to solve this problem. They put me on the spot. What can we do to solve the problem? You are asking, can we put more chemicals in the air, right? To kind of allow the ozone to go away or the hydroxyl radical to be tied with, or the molecules rather than with the floral scents, right?
My question is more, so if like in your experiment, instead of the insect being exposed to ozone plus linalool or one of the other chemicals, so it's reduced, but instead of that, keep the same concentration of the target chemical, but then add something else to it. And I just use benzene because it's a common pollutant that you would see in an urban area. So.
Yeah. So I need to think about it. But what I was going to say is that I would not propose to add more chemicals to our environment. What I would then propose, and to finish up the story of the New Jersey high school students, is that I told them, why do you plant flowers that produce molecules that are not readily oxidized, such as the petunia flowers, for instance? And that's one way. And believe it or not, I thought that it was kind of a crazy idea. The students, a few weeks later, came back. They thanked me and said, oh, this is a brilliant idea. I didn't think it was. But there was a science article that came out in July or August this year where someone actually posed the same question. What can we do for our pollinators? And then there was an editorial piece in which they said, well, if we go to places such as Iowa, Nebraska, perhaps instead of using the whole landscape to cultivate soybeans and maize, perhaps we can leave, some land where we can cultivate the flowers so that the insects can actually come and do their things. So those are the approaches probably that we need to consider. Again, I'm not a big fan of adding more chemicals to the environment, but I think more flowers probably would be good in my opinion. Yes.
So along those same lines, this is just anecdotal, but in Paris, I think they produce honey in the city. It's actually really good because there's more variety of vegetation than in these big fields that only cultivate one crop.
Yes. Yes. And I don't know whether you're aware that in some places, in the big cities, the greening of our urban environment is just catching on with popularity. And I was based in a town in New Jersey, where all the houses are decorated with houses. There is a profile of the flower beds in those houses. Oh my goodness, you see the bumblebees, happy dancing around the flowers. I think that's a good way to kind of help a little bit about the insects. I know someone else has a question.
Actually, very nice talk. I have several questions.
Yes.
First, the study that you did down, did you test to see whether the beetle can learn? In other words, if it accidentally goes down the right tube and then you reintroduce it, does it then select the proper tube?
Yeah, very good question. Yes, we tried that, we tested that, and I'd like to repeat what you just said. When you give rewards to the insects, they learn. If you have a hive of bumblebees, and if you put flowers, they learn, how to go and tell their workers, go this way, and they find it.
That's my next series of questions.
Yes.
And it begins with, how far does a honeybee travel look when it's foraging? So in your example, in your modeling case, you had the fragrance from the target enveloping the hive.
Yes.
But my understanding is that the honeybees travel much greater foraging distances than the fragrance from the target can reach.
Yeah. The honeybees have a foraging range, a radius of 800 meters. The bumblebees have a 1,200 meters radius. And they can only fly, most of them fly up to 8 to 10 meters. They don't venture higher. And they are also very good at meteorology because they don't venture out when the wind speed is higher than 5 meters per second. So they always stay home, when the wind speed is higher than 5 meters per second. Yeah.
Can I just interject 1 second? So if you have a hive, a single bee keeps within 800 meters a radius of that in their lifetime?
When they do their reconnaissance, because they do that reconnaissance first, they do that. But once they find the nectar and the pollen, that's when most of them fly. These are the natural bees, not the ones that we like to keep in boxes. They basically have a radius of about 300 meters on average. Yeah.
That's surprising because you see a lot of flowers that don't have any obvious hives within a kilometer.
Oh yeah. Not all flowers are pollinated for that reason. Yeah.
So my next question is about the social aspect. My understanding is that one bee will go out foraging and then discover a target area, and then will come back and communicate to the other worker bees in the hive.
Yeah.
And then they all go out.
Yeah.
Is that
We did not include that feature in our model, but that's how the bees actually do their things. In fact, when the bees, they have my colleague who is a collaborator, he has done all that work. When they know that there is a very rich environment, they have different ways of expressing that. One of them is they do the dancing, they dance, eh? Oh, can you believe that? They do the waltzing and all of that, the Macarena and all of that. It's interesting. Okay.
So then the last question is, do the bees or other insects remember where the target area is from year to year?
I asked that question a few months ago, and I did not get the answer for that. Yeah, I do not know.
So that'd be like pressing on generational knowledge.
We'll see if they can learn.
Yeah.
So the idea, and then I guess I did have one more question. I've heard that the alien population has started to recover only within the last, say, 2 to 3 years.
Mm-hmm. The what population?
The bee population.
The bee population. In some places, yes, they have, but in other places, they haven't. I do a little bit of research in the Florida Everglades. Last year, they got killed. 2012, all the bees that went from New England to Florida, because in 2012 we had a very major cold spell in Florida, they were wiped out. Yeah. So I think for the commercial hives, we have statistics, but for the natural hives, we don't. In the United States, we don't do a good job of monitoring the colonies of our insects.
I believe that the European Union has been pretty much.
In England, in the Netherlands, they have programs to monitor the population of the bees, but not in the United States. Yeah. Someone raised their hand back there, I think.
Oh, I guess I was going to ask about the strength of the evidence linking ozone and concentrations to plants and the pollinators. Ozone is going up some places, going down in other places. If ozone were the major factor, then you would expect pollinator populations to follow those trends as well. But, I'm sure there are so many other factors.
Yeah.
So what's your feeling about the actual evidence out there for the ozone pollinator connection? Or conversely, how big a factor do you think ozone is compared to pesticides, diseases, and other things?
No, I would say to you that based on that article that came out in Science last December, pesticides win the prize in terms of killing the bees. I'm not saying that pollution kills bees. I'm telling you here today that pollution confuses the bees. I have used that word, confuses the bees. And there is...
Meteorology to show that?
As a matter of fact, yes. There was a study that came out this past October. A colleague in Germany did a study as to what the threshold level is of ozone that will impair the neuroreceptors that the bees have the antenna, the neuroreceptors that basically they use to basically latch on the scent molecules. And that number is 1 parts per billion of ozone.
1 part
Per million, yes. So, but I want to also say to you, and in connection to this question, that we do not have any of the data to relate the long-term trends of air pollution and the health or the rate of confusion of our insects. We just don't have the data. And there are 3 people in the world who are nibbling at this research question. There's one group in England, in the Netherlands, and there is another group in Finland, and then me, the geezer here. Yeah. So we need to do a lot of work. And I said to you, I gave you this slide where I rank, what the key factors are that we know for a fact that are killing and destroying our bee pollinators. And then I gave you the highlighted piece that the pollution is one stress. I did not say that pollution kills the bees. But although I can give you evidence that in places where we have extreme levels of pollution, some of the pollinators are not there. But we don't have the evidence to say that pollution killed them all. Yes?
So maybe a better indicator of the effect of this is just the crop health, which we know has an ozone.
Yeah, that which it goes back to that magic number of 80 parts per billion, although there are some crops, such as fava beans, very sensitive to ozone. If you fumigate those plants with 60 parts per billion of ozone, you will, create, you will damage the stomatal pores of those plants. Yeah. Jeff, you have a question.
Yeah, one question, one common question. The question is, what is the flight pattern of the bee when it's foraging? What I want to know, does it sense the wind direction and then it go in a zigzaggy path to locate a plume, or is it just some random pattern?
They follow something that ecologists call the Levi random walk. So they follow a random pattern, and that's the model I used in this study here. And they are always, instead of following the wind, they go with the gradients of the concentrations. So they are probably very good chemists, because if, to have only 6 molecules impinging on their antenna, that's how they can actually go and follow the plume.
So are you lucky then that they find this gradient from the pollution and they swing right up it?
No, they keep.
Which is going to increase the gradient.
Because remember, I don't have a picture to show you. If I have pictures to show you, that when you create these plumes of these scents, you see clearly the filaments. These are the filaments created with the large area simulation. So you have one streak of highly concentrated scents, nothing in between, another one over there. So these insects probably, are doing something to navigate, to learn where the strongest gradient is. But I have to tell you that what I'm just saying here is based on what we think is happening. We do not have the information. And John Orlando and I have spent, and also Jeff Tyndall, part of this fall trying to come up with experimental protocols in which we go and fumigate these flowers, and then we determine the progression of the plume, and then we have two radars, one downwind, the other upwind, and we ask the bees, go and find the flowers in this environmentally modified landscape. And that will be the first data set to answer these questions you have. We don't have those data sets. Yeah. So we are, I think this is a very rich area, to explore all of these ideas. Yes?
In your numerical study, the control had the fragrances enveloping the hive. So the bees leaving that hive would not be following a random walk towards the target. Because they can sense the concentration gradient, so they should be following.
No. In the model simulations we did, they didn't. The hive was not within the plume of the sense.
I thought your control covered that.
The control.
Yeah, in the picture.
Oh, the control you mean?
You had the fragrance covering the hive.
Okay.
Control. So they're not following
Yeah.
a random walk.
The control probably, though you're right. The control, yes, but the other one's not.
So the implication of that is the hive is always located within the
Yeah.
origin distance
Yeah.
from the target.
Yeah.
So they never have to
Yeah.
undertake a random
Yeah.
walk.
Yeah, and that's a good point.
But
Yeah.
that's inconsistent with the idea that they forage based on the random walk. Right?
Initially,
They're
for
always within the fragrance
Yeah.
distance.
Yeah.
They don't have to
Yeah.
do a random walk.
Yeah, for that particular case.
Specific molecule. There are things that don't reach a number.
Well,
There's some stuff that only makes it 15 to 40 years.
So
Yeah.
maybe you're locating the hive.
Yeah, 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 yeah. That's a good point. Yeah, for the control experiment. Yeah, very good point.
But I mean, that's the way all hives are, right? Because they're within a kilometer of, they forage no greater than a kilometer radius.
Yeah,
So they have to be finding
yeah, yeah,
the nectar within a kilometer.
yeah.
Or
Yeah.
so.
Yeah, but there is one there is something incompatible, between what happens in the real world and what we include in models. In models, we assume that the wind constantly flows in a uniform direction, and that you must probably admit that seldom happens in the environment. The wind is moving randomly, and for that reason, I think in some instances they have to use these random walk simulations.
So I mean by distribution, just a binomial with a flat tail, right?
Yeah.
If I remember correctly.
Yeah,
Just
yeah, yeah,
More likely than
Yeah.
binomial to have outliers.
yeah. Jeff?
Yeah, we've talked about this before, but the diffusion that you're using there, what we would term mean diffusion, Taylor diffusion,
Yeah.
particle diffusion. And I think what you want to be looking at, if you're talking about short time scales and length scales, is the relative diffusion about some meandering plume as it
Yeah.
moves away from the source.
Yeah.
Because bees are operating on presumably short time scales, and they can see a much higher, or sense a much higher concentration than you would get out of
Yeah.
modeling mean
Yeah, yeah.
diffusion.
Yeah, I think in the Lagrangian model, that's a feature that has to be modified. Yeah, in the large area simulation, I think that's already included in there.
No, it isn't.
No?
No.
No, but we put the, let's see. So in your case, you're suggesting that we really go to the size of the bees. That would be the scales of the turbulence that we have to resolve.
Something maybe a little bit bigger than that, but
In that order.
it starts out of that order.
Order, yeah.
Increase.
Yes. Yeah.
Because what is the source size in here? Is it a grid cell at the LES?
We, in the LES, we put squares, yes.
Yeah, but what is the size?
Oh, in this case, we planted basically an area of,
Grid cell.
The grid size is 10 meters. We use 10 meters. 10 meters. Yeah, that's right.
The source can be much smaller.
Yes, you're right. Yes.
Have a much narrower plume.
Yes. Yes, you're right. Yes.
At the 1 or a few centimeter scale, we start fixing puberty. With molecular behavior, but that's
Yeah, but with the wind carrying its dissipation rate, which spreads its plume, which initially grows as distance to the 3/2 or
Yes.
100 to 3/2. Different pattern than what you have with tailor diffusion.
Yes. Yes. Yeah. The B, that's right. Yeah. 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 Yeah.
All right, so